want to miss that. Share the screen and the PowerPoint. We should be ready to roll. Maybe. There it is. Anaerobes part 2. Okay, so we know we've already looked. Some of these names are just they're going to be adventures. I can say that already. But you know, I've been preparing you all semester for this one, right? So these are our non-spore forming gram-positive rods, which that means part 1 at our spore forming gram-positive rods. A little different take today. I think I can get the first part. I think everybody. Can y'all get the first part? I'm going to go with bi instead of bi. So I'm going to go bifido. Bifidobacterium. Eric? Sony. How about that? Is that good? Bifidobacterium Ericksoni. Unless y'all have a different way. I'm open to anybody that wants to jump out there and correct my pronunciations and let me know I need to be better. So again, non-spore forming gram-positive rod, habitat, non-pathogen from oral or intestinal mucosa. Morphology is pleomorphic gram-positive rod. And then we're going to spend a little time on biochemical identification. So it was brought up to my attention last lecture about the catalase reaction. And it was pointed out to me that they saw a catalase positive on the anaerobic side. And I said, yes, it could be there, definitely. But we're going to see this right now as catalase negative for the bifidobacterium. Indole negative, unlike our one of ours we have downstairs for today on our gram-negative panel. Nitrate negative. That's very unusual too because most of our organisms we pick up in the lab are nitrate positive. Escalin positive. The patterns of acid produced in peptone yeast extract glucose broth can be used to differentiate the anaerobes. So we can do this peptone yeast extract glucose broth. The major acid for Bifidobacterium Ericksoni is lactate acetate. Lactic and acetic. So lactate and acetate, major acids. Did y'all learn that in chemistry, that 8 at the end means acid? No? Yes, good. I was hoping you picked something up on your chemistry. Here's our gram-positive rod again. If we see gram-positive rods, it's always a shocker because we're not expecting gram-positive rods in anything that we do. And then they're hard to ID, so we reference a lot of them out. So we will see this from time to time. Okay, how about that? Spent a lot of time on that one. So we're kind of introducing them, and then we'll spend a little time talking about them. How about this one?
That one is not.
You don't have this on your slide?
No,
Cutie's not there on yours?
no, we go right to.
Oh my goodness.
something.
Okay, sorry, this wasn't in the student.
Well, no, it is that. We just don't have the new name. We have.
Oh, y'all have the formally, yes. So
Yeah.
that makes it more fun then, right? So yes, what I gave you is I give you the student PowerPoint that I have without, like, probably maybe hadn't been updated since we did this update. So we have Cutie. Are we gonna go with Cutie? Cutibacterium. Cutie, right? Cutie.
It says cootie, like.
Cootie, you're going cootie.
I mean, because the in parentheses.
You want to go with cootie?
Yeah.
Okay, Cutibacterium.
I'd be able to remember it easier.
We going with acnes?
Yeah.
Okay. Formerly known as, we don't really need to know, or do we need to know formally?
What's on your poll?
Yes, I think so. Propioni. See, I like that one better. Propioni bacterium acnes. Now we're going cootie bacterium. Getting the cooties. See, that's what I think of cooties. These aren't cooties, right? What are cooties? If y'all never heard that term before, cootie,
Well,
cooties?
yeah.
Oh, yeah, we
You're gonna get the cooties
As a child.
as a child. What did it mean when you were a child?
Don't kiss
Kissing
boys.
each other, you get cooties and kid. There you go. Yes, exactly. Right? Okay. Good. That's great. Thanks. So this is a gram-positive anaerobic rod, which is so named for its casual association with acne vulgaris. It is a normal commensal of the human skin. It can cause a wide variety of diseases such as central nervous system shunt infections, joint infections, endocarditis, and osteomyelitis. So it's not a, non-pathogenic. So, but it is part of our normal human skin. So hopefully you can remember that, if you're kissing little boys when you were younger or kissing girls when you were younger, you were told you were going to get cooties. All right? Thank you. Glad you have heard that. I think y'all are just all too embarrassed to talk about it. It frequently contaminates our blood cultures. So yes, so if we get a gram-positive rod in our blood culture, could be because we'd had phlebotomist or a nurse or EMT or a doctor or a medical lab scientist that didn't know how to prep the arm before sticking for blood cultures, and we end up with a contaminant. Morphology, pleomorphic gram-positive rod with a beaded appearance. So that comes up quite often with beaded. You'll hear that. Diphtheroids. But I think I saw I have friends on Twitter and Facebook that post a lot of micro stuff, and I don't know if y'all follow me or not. But it's a good way to catch up and get a little quiz every once in a while. And I think this one popped up the other day where somebody said a beaded appearance gram-positive rod, I think came up. So definitely would send you to the Cutibacterium acnes. Biochemical identification, it's a switch, right? This is one of the ones that I think was brought to my attention the other day. I said, look, Mr. Director, I'm looking up here at wherever we were. I don't even remember where we were. One of the gram-positive rods that was anaerobic, and they said, hey, it says it's catalase positive. And I thought you said they were all catalase negative. That's what made them anaerobic. This would be a little different, right? Catalase positive, endo positive, nitrate positive, escalin negative. So it's just opposite of what we just saw with who? Who was the one we just saw? I can't hear. What is Ericksoni? What was the first part?
I don't know how to say it.
We'll try. That's why we do this. This is we're all amongst micro friends, right?
Okay.
B foreign to bacteria.
Okay, I just went with Fido. Like, buy Fido. That's easy for me. Like, Fido's a dog, right? That Fido. Y'all ever hear a dog named Fido? Is that a book? Fido. Buy Fido, Ericksoni, right? Good. Thanks for playing along. Right opposite, right? Because it was just right opposite because it was catalase negative, indole negative, nitrate negative, but escalin positive. So I think I want to check, make sure, but I think you had these on the back of your thing. I think it's on there. Yeah. It's a little small to read, but I think it's on your PowerPoint a lot better. Let's see. So propionic and acetic are the major acids. There it is, gram-positive, pleomorphic. So not that distinct, spore-forming, boxcar, parallel rods. These have got a little bit of movement and change and, as we say, a little flexibility. That's what makes them pleomorphic. Okay, like I'm looking here, that, we're looking there, that looks like the seagull wings from way back that we talked about. Ooh, scary there, right? All right, that's 2 down. Okay. We going with egg or egg? Which one are we gonna go with? Egger? Egerthella? Lenta? Or are we going with eager? Eager. Egg. I like egg. It makes me think I gotta say 2 G's. Spell it with 2 G's. Egger. Egerthella lenta. Habitat, oral and intestinal mucosa. Implicated in the cause of ulcerative colitis, liver and anal abscesses, and systemic bacteremia. Egerthella lenta. Remember, gram-positive rod, non-spore forming. So we kind of keep this going together. Biochemically inert. Who else is that? Who else is biochemically inert? Shigella. Yes. Shigella is not an anaerobe, right? So we would definitely connect this one with a biochemically inert anaerobe. Catalase negative, indole negative, nitrate negative, esculin negative, unlike our biphyto that we had earlier. And the major acids are butyric and acetic acid production. There it is. Definitely has some wings in some of those. And then we come to a more friendlier, sometimes, not all the time, but it is. I think we can just say yes. Because we talked a little bit about this yesterday in our lab for those that were there. We brought up lactobacillus. But for us and for you, predominantly the organism in the vaginal tract, lactobacillus. This genus contains anaerobes and microaerophilic. So tell me something about microaerophilics again, because we know anaerobes. What's microaerophilic mean? Remember that was back in the lecture on Tuesday? What's microaerophilic? Zoom, y'all can talk. I can hear you this time, so if y'all want to chime in. They're waiting on you to chime in because they're all looking like I'm not going to answer in here. Microaerophilic. Small something, right? Small air, small oxygen.
It's an organism that lives in an environment that only has a low level of oxygen.
Okay, say it one more time. I had to turn you up a little bit. Go ahead.
Okay, it's an organism that lives in an environment that only has a low level of oxygen.
So it can tolerate a little bit of oxygen?
Yes.
Okay. Morphology, gram-positive rod, single or in chains. We do see chains. Biochemical, it looks inert too. Catalase negative, indole negative, nitrate negative, and variable esculin. We always love when they put variable esculin. And the major acid is lactic only, lactic acid, lactobacillus. That's how it gets its name, lactobacillus. Clinical significance is that as lactobacillus, we said it was part of what? Part of the vaginal flora, basically. Certain species of lactobacillus, especially those which can be vancomycin resistant, occasionally cause opportunistic infections of normally sterile sites. Lactobacillus can also cause UTIs. They appear to be exceedingly rare, but Lactobacillus delbrueckii can be probable agent in UTI in elderly women. How we doing? So far, so good?
Great.
So far, so good. Goodness. If we have yeah, we have lactobacillus. So on the rods here, under the catalase negative rods, we had lactobacillus show up. Yay, fine. We also have actinomyces and Arachnia species. Actinomyces and Arachnia. Actinomycosis and actomycotic mycetoma. Those are the diseases caused by actinomyces and Arachnia. Habitat, colonized oral cavity, and the female genital tract. I see actinomyces here, but I don't see Arachnia there on your little shortcut sheet. Actinomycotic lesions. Actinomycotic lesions. Here's the one on your short list that we have on our little our flow chart. Actinomyces Israeli, or Israeli. How about that? Is that better? Israeli?
Yeah.
Okay. Actinomyces Israeli. Rough heat molar tooth colonies. Molar tooth. So who's my pre-dental students? Do we have any pre-dental students? You know what a molar looks like? What's a molar look like?
It's like the ones back here.
The back there, so like squared off kind of.
Yeah.
Is that the molars? Okay. Microscopic, long branching, gram-positive filaments, rods, cocci. Got all kinds of good stuff with this one, right? Actinomyces. Smooth, flat, or convex colony, branching gram-positive rods, diphtheroid forms, and there we go. Molar tooth, looks like a tooth, right? If you're looking from the top on the tooth, right? If you open your mouth, right? Looked at your molars. Hope you can see that. Kind of got that. Or as my dentist likes to say, that's where your cavity is going to be. Like, puts that probe right in there and goes, it's not soft enough for a cavity yet. There's our wild gram stain. Okay. Filamentous, chainy, clumpy, just running all over the place kind of growth. That. So this is one, when we think of, I guess where it gets its kind of maybe it's arachnoid look. Biochemical identification for actinomyces and Arachnia is catalase negative, indole negative, nitrate positive, escalin positive, so a little twist there. And then the major acid is lactic and acetic. So here's one of our biochemical ID charts that sums up everything that we've talked about so far. Gram-positive rod. We got our Bifidobacterium ericksoni. We got our Propionibacterium acnes, which we renamed Cutibacterium, right? Cutie. Will we always see it one way or the other? Don't know, right? I was talking to some students earlier today, and they're like, well, we hate looking at old textbooks, and, there's stuff they'll say, oh, we don't really need to know. You just never know. Like I said, there are humans that write these questions on the board for you. These are old teachers that could be still in propionic bacterium and hadn't updated it to Cutibacterium. So definitely could see it either way.
For the actinomyces, it says on our paper, lactic and acidic or whatever,
Yeah, I was
and that
about to
says something
ask.
different.
It says succinic, doesn't it?
So,
So what do we need to do? we're gonna have to refer to our text. All right, anybody following along?
What page?
Oh, that's what I'm looking, where are we in chapter 41 or chapter 42? Let's look, see if we can, see if our textbooks actually helps us out here. There's a whole list there of Clostridium. Move on. Bacilli. We're not into Mycobacterium, so we must have gone past it. Index, right? I think I was in the right chapter, but we'll go back. Which one is it? Actino? Let's see. Act.
It's on page 519.
I was right there. Okay. Does that tell us which acid? I see sulfur granules.
Yeah, I'm not seeing.
Oh, I thought you had it. You were like,
No.
contain, there's gotta be some. I got my hat on. I'm having a hard time finding it in the book, aren't I? There another sort. Where else do we have that coming up? I was thinking y'all had it.
Google it. Put it on other items.
Google's not much help either. It just says
Oh.
that they're anaerobic.
Gosh. What is succinic acid? Maybe that'll help.
It's a drug used for nutritional supplementation.
That doesn't sound like it would be producing that. Succinic. I see sulfur. That's why I was thinking maybe they've connected sulfur. Good catch. I'll mark it and see what I want to say. I'll say it in lab when I get down there. Okay. Everything else okay with the major acid produced? Succinate, wouldn't it be under succinate, being the acid? What's different from succinate and lactate? We'll run that one through the chemistry department. Zoom, you got an opinion?
All it says is that succinate is used for lactic acidosis.
So we do have a relationship of some kind. I didn't know if one was before the other. We'll come back to it. All right. Anaerobic gram-negative rods, is that where we are? Okay, so general characteristics. So now we've kind of introduced, I think the way you want to look at this is we've introduced the players. So now we're going to kind of talk a little bit about in general. Atmospheric requirement, moderately obligate anaerobe. Makes sense, right? Means they can do what? Survive a little oxygen, right? You don't have to, oh my gosh, we might have an anaerobe. We don't need to let oxygen get here because it's going to die if it gets in front of it. Habitats, colonized mucosal surfaces. I think we got that set up where it's either the oral, intestinal, or the urogenital tracts. Anaerobic gram-negative rods are anaerobes most commonly found in anaerobic infections. Yay, we're happy about that. They usually results from trauma, surgery, diseases which damage the mucosal surface that allow those organisms to be introduced where they're not normal. Okay, so we think we have that already established. Infections usually characterized by abscess formation and tissue destruction. So once they come off the mucosal lining, you know, when we think about oral, what happens there, right? Dentist work, introducing those into areas maybe they're not supposed to be, like your bloodstream. We also think about bites. When somebody bites, a human bite, supposedly, you know, awful, awful, awful. Because then these get introduced into areas they shouldn't be in. So site of infection, it can be aspirate pneumonia, which is never good, right? What is aspiration pneumonia? What has happened? So this can be as simple as they were under anesthesia and they forgot not to eat before they went into surgery and they vomit during surgery, right? And you suck vomit into your airway. That's one way to aspirate into a pneumonia. Or maybe this one sounds more closer to home. Our buddies are so drunk that they throw up while they're passed out, and they actually what? Aspirate, right? They actually pull vomit back into their airways. That's another good way. Okay, that can happen. So, or we're in a nursing home and we're not up at an incline when we eat, and we actually throw up a little bit, and they've got our beds are so far down that we end up aspirating again. So we see this in the hospitals. We see them from the nursing homes where they have aspiration pneumonia. That is one way. So that would be inside the lung, setting up an infection where it doesn't normally be. You get a sputum culture, you end up with gram-positive rod, right? Not TB and not normal flora. You would end up with a gram-positive rod infection. Intra-abdominal, the result of a contamination of peritoneal cavity with bile contents. So that's trauma, surgery, appendicitis, inflammatory bowel disease, colon cancer. All of those can exit the bile. And remember that the bile is external. That just because it's inside your body cavity doesn't mean that it's internal to the body. So that's still external. So any perforation, any ulceration, any surgery where they're cutting that open, you really got to be careful that you're not sending some of these into the abdomen. Female genital tract infection, post-abortal sepsis, pelvic abscesses, endometriosis, post-operative infections, intrauterine devices, anything that is leading that mucosal into areas where it doesn't need to be, and set up these infections. All right. We do have this one on our list, I think, didn't we? Didn't we have Bacteroides fragilis? Did I not see it on our little chart? Did I miss it? Or am I just dreaming up that it's here? Maybe I'm dreaming it's here, but I thought I had Bacteroides fragilis on here. Anyway, another group, Bacteroides fragilis, and we have a whole list. So we have Bacteroides, Dista sonus, going with that, Dista. Ovatus, Theta, Iota, Omicron. How about that one? Vulgatus, Uniformis. So we usually, if you take the top and the bottom, those are the ones I've usually have heard of more than anything else. It's usually Fragilis and Uniformis. The nomenclature change of Bacteroides distasonus to Parabacteroides distasonus renders the Bacteroides fragilis group terminology no longer up to date. Clinical Lab Standards Institute has changed the text from Bacteroides fragilis group to Bacteroides species and Parabacteroides species where appropriate. So I hope this doesn't surprise you that microbiologists have decided to get together and decided we need to make a name change. They seem to do that quite often. So every couple of years, something changes, and we as faculty and you as students get to suffer from it because we know it one way and then it gets changed to another way. Same thing as we did with Clostridium difficile. Now it's Clostridioides difficile, right? Still C. diff, still the same organism. Just needed to make a name change. We didn't want to go too far and confuse everybody. So here again, we're changing up some species instead of a Fragileus group. Is this in our book? Is anybody following along?
Yeah, it's on page 520.
520, good. What do they say? This is supposed to be, I mean, there's a new edition coming out of this,
Yeah,
but
they're still calling it
group,
Fragilis.
yeah, so these are our experts that we rely on. They're still calling it group, so I'll still hang in there, but this is letting us know that there has been an update change. So Bacteroides fragilis group, microscopic morphology is gram-negative Coccobacilli. So gram-positive rod is still there, but we get a little bit of change with the shape. Somebody chiming in, Lauren?
Yeah, sorry.
Okay, what'd you want?
No, my stuff died again, so I had to come back.
Okay, welcome back. Bioresistance, bacteria grow in media containing 20% bile. So we're going to increase the bile of the agar, of the media, and Bacteroides can thrive in that where other organisms may not. Resistant to aminoglycoside antibiotics, and of course we're coming up to that, so that's kind of one of those, maybe you like file back a little bit so when we get to the antibiotics you'll know what aminoglycoside is and used for. We did have this on there, right? Didn't I see it? Where is it? Bacteroides? I thought I've seen this on our sheet. There it is, way over here. Right over to the right, sorry. Gram-negative rod, way over here. Everybody see it? It's right under Helicobacter pylori. We're over here with Bacteroides fragilis. Anaerobe, colonic flora, abdomen pelvic infections, brain abscesses, bipolar staining, and beta-lactamase producer. Yay, we found it. I found it. Y'all were supposed to help me with that. So Bacteroides fragilis group, indole negative for fragilis, catalase positive, ovatus is positive indole, catalase negative, and theta iota micron is indole positive, catalase positive. Again, gram-negative rods in our anaerobic section. That's straight. Biosensitive, pigmented, gram-negative bacilli. Well, what's the difference here with biosensitivity and bioresistant, right? Isn't that what we call fragilis? Bioresistance. That's key to remember that Bacteroides fragilis group are bioresistant, meaning they can withstand bile. Our Prevotella melaninogenica. How about that? I think I did really good on that one. Melanino, did I do good? Yes? Y'all should be, I'm like getting so much better through the semester. But is it Prevo? It's Prevo. We're gonna go with Prevotella melaninogenica. genica, and Prevotella intermedia. These they colonize the oral again, gastrointestinal again, and urogenital again. So this is kind of the running theme of our what? Anaerobic rods, whether they be gram-positive or gram-negative. These are pigmented. Not only are they bile sensitive, unlike our Bacteroides group, right? They're also pigmented gram-negative rods. Oh, and they give another one right off here. Let's see, Porphyromonas, Porpyro, is that where we're going for? Pyro, not pyro, Pyro, Por. Okay, Cadis, it's your chance. Go all the way through. I want to go to the sacral, whatever, too. Go.
Porphyromonas.
Porphyromonas, very good. Asaccharolytica. Yes, yeah. Yeah, it colonizes the GI and urogenital mucosa, but not the oral cavity. We have a different location for our Porphyromonas versus our Prevotella. These organisms produce a tan to black colony on the anaerobic blood agar plate. Now, quick quiz, what's in that anaerobic blood agar plate that makes an anaerobic blood agar plate? What's in there? What did we add to that blood agar?
Vitamin K.
Vitamin K and hemin. Yay. You are on top. When you go to microclinical and you throw that out there, it's going to be very impressive. When you say, oh, I know what's in the anaerobic blood agar plate. Wound exudates and colonies growing on media exhibit a brick red fluorescent. So not only growing, but actually inside the wound, okay? The oozing from the wound will have a brick red fluorescence look to it. Ugh. So they produce tan and black colonies on the blood agar plate themselves, but they will have this brick red fluorescence too. So again, we can check indole, also check lipase. Prevotella intermedia is indole positive, lipase positive. Prevotella melaninogenica is neither, and Porphyromonas asaccharolytica is indole but not lipase. So we have that distinctions that we can make biochemically. And hopefully it's focused on the major differences that you can remember. Okay, so that's why when we did lab, I was like, oh, you can use this, help you out, help you remember some of this, but you don't get to use this on the test. So these kind of things are what love test questions to come up with and use. So those were our bile-sensitive pigmented gram-negative bacilli. And now we're at bile-sensitive non-pigmented gram-negative bacilli. So what's the key problem right now? If y'all picked up on one? You know, we said gram-positive rods, we're going to see those. They're going to be, scary. What about gram-negative rods? Who are our gram-negative rods on the aerobic side? Just so we're thinking on the same path here. Who are they? E. coli, Klebsiella, Pseudomonas, right? They're going to be gram-negative rods on gram stain. Okay, these are anaerobic. These are, where are your genital tract? Intestinal mucosa, oral cavities, right? I mean, I'm not thinking E. coli and oral cavities, but thinking urogenital, yeah, we can find urogenital E. coli. We can find definitely intestinal mucosa with the enterics, right? So don't forget that we got other gram negatives in this area, okay? The bile-sensitive, non-pigmented gram-negative rods. We got one. Prevotella again. Aureus is here, and Prevotella. Who? Okay, to tell me out with Bucky there, but.
Oris,
Huh?
Oris,
Oh, I got Oris right. I was thinking of the other one.
oh, that.
Yeah. How are we gonna go with the
Why does
second?
it have a dash?
It just probably has some more
Oh, okay.
names. Y'all were supposed to put something in there, but we don't have it.
I don't know.
View, okay, good buck. Okay, Bootsy. How are we gonna say that last one?
UK sounds better.
UK. UK. Okay. These are infections of the head, neck, and oral cavities, upper respiratory tract, genital tracts, bite wounds. Okay, so we definitely are thinking along the bite when we talked about the human bite earlier. Bile sensitive, they do not grow in the presence of that 20% increase in bile. How could we do that? Do we really need a, how are we gonna have auger here? Are we gonna have plates for this bile sensitivity? Has anybody ever heard of something we use, one of our growth medias that we like to put these in? Just to,
Bile salts?
bioesculin. Okay, so we'll have bioesculin available for this, right? We'd increase our bile content, would definitely help us here. These will not grow there. Non-pigmented habitat oral cavity. So Prevotella aureus, which makes sense, and buccal is why I was wanting to go buccal, because I think buccal cavity, you know, the mouth. But I'll go with Buccae. What did you say?
I said UK.
Buccae. Okay, Buccae. All right, we'll go with Buccae. All right, this is another one on our list at the bottom of our gram-negative rods. This is our Fusobacterium. Very long tapered rod, it says here on our little reference sheet. This is Fusobacterium nucleatum, and Fusobacterium necrophorum, the most frequently isolated, most pathogenic members of the genus Fusobacterium. Nucleatum is the most common isolate in the head, neck, and lower respiratory tract infections. Again, these are gram-negative rod. Necrophorum is the most virulent. It may invade the bloodstream following tonsillitis to cause septicemia and widespread abscesses. Habitat, mucosal surfaces has been our common theme pretty much all throughout the day. They colonize the upper respiratory, gastrointestinal, and the urogenital tracts, kind of our theme of the day. Their microscopic morphology, very long, thin, fusiform gram-negatives with tapered ends, rods with tapered ends. Means they're coming to a point, to the end, okay, tapered. Necrophorum is pleomorphic gram-negative rod, meaning it can take, it's flexible. So there we go. There's our nucleatum. Necrophorum, a little different. Doesn't have the tapered ends, but our gram-negative rods. Got a lot of different moving here. They're non-hemolytic. So already what changes there? What is E. coli? Often beta hemolytic. That'd be a quick way to judge that, wouldn't it? But often show a greening of the auger when exposure to air. So they almost have an alpha hemolytic look, Fusobacs. The nucleatum have speckled colonies, and the necrophorum, which is very, very, very important description here, fried egg appearance, okay? Necrophorum. Again, anaerobic blood agar plate. I think we saw a picture of him earlier when we were talking about the media. There's the fried egg, look. Y'all know fried eggs? Is that a term you still understand, fried eggs?
Yeah.
Yes, y'all know you can fry egg. You don't have to scramble all of them. I didn't know. I didn't know where we were with the diets of the new generation.
Yes,
Not
let's go
eggs.
fry
Because some kids don't think the eggs only come scrambled, right? That you always have to have scrambled eggs.
Who
That's-
thinks that?
my
Yeah.
kids do.
Do you not make them other eggs?
Yeah, my daughter, I started making her fried eggs this past summer, yeah. But yeah, because when I was a kid, my dad, like, that's all he ate was fried eggs. Like, fried egg every morning, 2 usually, all right? So I saw it every morning, but I didn't like fried eggs. I don't want scrambled eggs. So I didn't know where y'all were with that. Because I don't get up every day and eat fried eggs like my dad. Sorry, I don't. I barely get anything. Fusobacterium species, Nucleatum and Necrophorum. The biochemical ID, I hope the major acid is butyric. We'll double check, all right, hopefully. Biosensitivity, meaning if I increase the bile content, they will not grow. Indole positive, esculin negative, and lipase positive for Necrophorum. I hope that came across. Succinate shows up again right here for the bacteroides. I don't even think we talked about its major acids, did we? Till we got here. Okay. Now, not to confuse you anymore, now you can see why we kind of saved this lecture for the last couple in bacteriology. Anaerobe gram-positive cocci. Now that would be like, how many gram-positive cocci do we have? Whole heap, right? Staph, strep, everything else right in between. So these are the anaerobe gram-positive cocci. So switch over, right? We go back over here to gram-positive cocci and see if we can pick up some of these. I don't know where we're going to go, but let's see. Again, skin and mucosal surfaces. Infection, most frequently respiratory tract and female genital tract. Damage to the mucosal surface. And to ID these, we're going to use gas chromatography. That's new. I don't think we've talked about gas chromatography. Does anybody know about gas chromatography? Have you ever heard of that? Explain.
Classes.
Huh?
I don't want to.
Some other class? Yeah, you were shaking. Yeah, you didn't know what I was going to say. Yeah, gas chromatography. What is that? In simple terms, you don't have to go into a whole lot. Huh? Use a gas to do what with? What do you? Gas chromatography. Somebody, come on. I got some students in the back that haven't contributed any information in weeks. So y'all come up with this. Gas chromatography, what is that?
Use gas to color it.
Gas to color it?
I don't know.
Any help? Adam or Zeke Chong back there? Y'all help us out with gas chromatography? No? I see laptops open. Help us out.
I found out what your succinic acid
Okay,
is.
we'll come back to succinic
But
acid in just a second. I gotta get gas chromatography done first.
I can give you the basic description that Google gave me.
That's good enough. That is perfect.
It is a common type of chromatography used in analytical chemistry for separating and analyzing compounds that can be vaporized without decomposition.
Yes, yes. And what we see with gas chromatography is usually a peak of some kind, right? The computer picks that up as it comes through. So it's kind of a detection at a certain movement. Does that sound right? Sound right at all? Analytical chemistry.
Crystals or anything like that?
Yeah, I think so.
trying to remember.
So it's kind of to see the content of what you got kind of thing.
Organic does that. in the lab.
Organic does that too? Okay. Okay, that's enough chemistry and stuff. We'll come back to micro. Are we at Parvomonas here? Is that all these or is that Peptostreptococcus? What is anaerobus here? Robius. We got a P in front of it. Are they all Parvomonias or Peptostreptos? Got one little section in the book. Like this is like one little page, like 522. We're like right here, like little bitty, like one little line.
Is it Chrono?
Proto? I don't think we've introduced it yet. That's why I came back to it. You're going with which one? You don't know?
I found Anaerococcus, but not.
Maybe we can get some help. No, that's it. That's it for gram-positive cocci.
It says,
Peptostreptococcus.
You think it's
it says Staphylococcus aureus sub, and then it has the anaerobe. The staph isn't a keyword.
no, it's not.
I'm not sure.
It's like the second paragraph. It says among anaerobic gram-positive cocci, the genera.
Oh, so you picked it where?
The second paragraph under anaerobic gram-positive.
Up here in the book?
Yeah.
Oh, okay. Yeah.
So it's got a bunch of names, not just the one.
Yeah. But we're gonna go with what? What do you want to go with? Pepto?
Sure.
Is that what you just said?
Yeah, Peptostreptococcus.
Peptostrepto. Do we even have this on our little sheet? Peptostrepto. If we're over in streps, let's Pyro, Streptogalactiae, Enterococcus, Group D, Bovis, nah. Anybody find them on our little sheet?
It has Parvomonas and Peptonophyllus too.
Oh, it does.
Those are the other keywords, I guess.
So is it Parvomonas, formerly Micromonas, micros, and Peptostreptococcus, micro? So kind of again, changing the names up on us, aren't they? That's the point I was trying to make. Do we have them on here? Anywhere? I see cootie up here, so cootie bacterial now.
The book is calling it Pepto.
Pepto, okay. Peptostreptococcus anaerobius, meaning an anaerobic gram-positive cocci. I'll go with that. I'm good with that. Uh-oh, look at there. Looks just like our gram stains we started with at the very beginning of the semester. Not quite, but stay with me. All right, anaerobic gram-negative cocci. Gram-negative cocci. Flip over. All right, gram-negative cocci. Who do we have any of those over here already? Gram-negative red. That's Kleb, Shigella. Do we have any gram-negative cocci? We have diplococci. Neisseria over there, right? Everything else is rod pretty much. So you can, the point being that these are around, but are we going to be honing in? Are we going to be IDing? We just need to know they are around. So if you do an anaerobic wound and all of a sudden you got growth on the anaerobic plate, simple as that, but nothing else is growing. You gram stain it, you see cocci, and you go, oh, that must be some kind of staph or strep. Now you hopefully won't make that mistake and you'll lean into a little bit more on your anaerobe and say, you know, this one just doesn't seem right. Maybe this is an anaerobe, okay? I don't want you to like, oh, I need to spend all my time. But what you're seeing is, what's our identification? Gas chromatography. Is this, are we going Vel? I love Vel. Velanella? Or Velanella? What are we gonna do? Vela? Vela?
Kinda catchy.
You like Vel? Well, how do you spell Vel? Vel in like Colorado is like Veil, right? Is that
That's
right?
how that's spelled.
Yeah, so I was gonna go Vel. Velanella? Velanella parvulum? Parvulum? There, gram-negative cocci. A Brucella auger. So we can see, I don't want you to think just because we need gas chromatography for ID, we do have growth. Okay? It's not growing in gas chromatography. We've got growth in front of us. We're just maybe gonna have to say it's an anaerobe. Right, send it. So here, we'll get into some identification of the anaerobes.
Put that S word on this chart too.
What's that?
That S word for actinomyces.
Oh, we were gonna come back to that. Succinate. Allison's gonna update us on succinate versus lactate.
They're pretty much the same thing, except succinate, whatever, is used mainly in your food industries. It's used as a sweetener,
Okay.
and so it's just another sugar that it can use according to the internet.
Okay. All right. Well, we'll note that and just kind of
It says it can sometimes have a sour taste, in case you were
sour
curious.
taste. Okay.
But it's used as a sweetener.
Is that or the food additive? Let's see what we got here. We got succinate. So we're gonna end with succinate being our actinomyces acid, right? I knew we had this somewhere. I'd seen this and I was like, well, we're not way up front. It's different. Or in the lit. Was it in the PowerPoint?
It was in the fill-ins and it said to put lactate in the fill-in.
Okay.
We'll just have to go to the beginning with the other word.
All right. So here it's succinate. We put them all together. Okay. Can we do catalase test? Audience, can we do catalase test? Can y'all do a catalase test? Yes, y'all can. Yay. So this is, can y'all do an indole test?
Yes.
Yes, you can. Yeah, so we can do some of these things. At least those two for, what's P. acnes again? That was the cootie. Y'all were supposed to say that's the one they renamed cootie. Cutibacter. Not propopony, what was it? Propione bacterium?
That sounds right.
Okay. Our gram-negative rod, that was our gram-positive rod. This is our gram-negative rod. So we get them all listed out and we get it summed up in things like catalase and indole and esculin and lipase and 20% bile growth and major acids. Pergelius, ovatus, theta, iota, micron, intermedia, melaninogenica. What's the P again there? So I've already forgot. Bacteroides is the first one. What's the P for the second group?
Peptostreptone? Or is that-
No, it's before Peptostrepto.
Is this the Pronovella species?
Provatella,
That.
Prevotella, Provatella. And then what is the necrophorium and the nucleadium down there? What's that F stand for?
Who's that?
Fusobacter. Yeah. Okay. So hopefully you've enjoyed this lecture. But that's it. So those were our anaerobes. And that is definitely a group you won't hear much about, but you don't need to forget about. Okay. That's the important part. This group. Now the group we had Tuesday definitely are gonna hear about a lot with that C. diff sitting there. All right, downstairs, we got negative panel IDs, and then we're gonna introduce sensitivity, which is we're gonna look at our gram-negative panel, we're gonna ID our organism, and then we're gonna look at our antibiotics and start to get some names introduced to us and how we read those. So it should be not a full class time, but it should be a good section of it. We'll see you at 12:30. See you later, Zoom.